So in today's lecture, we're going to talk about how genes are controlled. Last time we talked about how we can take genes that are on the DNA, transcribe them into RNA, and then turn the RNA into protein via translation. But this whole process is very tightly regulated, and the different genes in different cells are going to kind of have different expression levels. So certain cells are going to have genes that are going to be transcribed and translated, and those same genes will maybe never be transcribed or translated in other cells. And so we're going to talk about that process today. So the first thing to understand is that every single cell in our body is going to come from a single original cell, which is going to be called the zygote. Okay, and so we'll write that out. So all of the cells in our bodies are produced through mitosis, starting as a zygote. Okay, and a zygote, this is probably something you've heard before, the word zygote, is the original cell that is formed after the fusion of sperm and egg. Okay, so when a sperm comes into contact with an egg, it then is going to form a zygote. So this could be our zygote. And then the zygote ultimately is going to go through a process where it starts to divide. There are different cell stage levels. So there's a time when there's like 2 cells, and then there'll be 4 cells, and then there'll be 8 cells, and so on and so forth, and things like that. And so that whole process is going to happen through mitosis. But one of the main things to realize about this is that the DNA, the chromosomes that came from the sperm and the egg are now together, and that DNA is going to remain the same in all of these different cells. So every single cell in your body is going to have the same DNA, which is really important. But then it's going to, we'll explain today how the different cells do different things if they all have the same DNA. Okay? So let me just write this out. So what this means is that every cell in your body has the same DNA as a zygote. Okay, and every body cell contains every gene. So if you think about this, we can talk about heart cells. Heart cells are going to have the genes that are important for producing the eye color that you have. However, they're not going to be expressed in those heart cells because they don't need to be. And the heart cells have to do different things. They're not going to be, pigment of your eye is not going to be important in that location. And so we're going to have lots of specialization of cells that's going to take place. Okay, so the cells in our bodies are going to be specialized in structure and function. Okay, and so we can, we're going to start ultimately out as a zygote, which is a cell that's not specialized in any way, and it's going to go through rounds and rounds of mitosis, and at some point we're going to start making different types of cells. And so we're going to make nerve cells, and so nerve cells kind of look like this. They have, these are going to be cells that are going to be important for, your sensations. We're going to have red blood cells. So these are going to be red blood cells. We call them RBC. This is a neuron. And then we can have muscle cells. So the easiest muscle cell for me to draw is going to be a smooth muscle cell. This kind of looks like an eye, but this is a smooth muscle. Smooth muscle is going to be found in a lot of our organs and our digestive system. They kind of help squeeze things and move things from one place to another. So the zygote is ultimately going to be able to produce these different types of cells, okay? And so they're going to become specialized. So the cells are going to have all the same genetic information, but they're not going to do the same things. And so we're going to start talking about how this is going to take place. Okay, so cells with the same genetic information, so all the same DNA, can develop into different types of cells through what we're going to call gene regulation. Okay? And so gene regulation is going to be a mechanism that turns on certain genes. And when it means turns on, it means that they're going to be transcribed and translated, while other genes remain turned off. Okay, and so, like I said before, in certain cells, the gene is there, but it's not necessary and therefore will not be turned on. So it's going to remain off and not be transcribed or translated. Okay? The regulating gene activity is what allows for specialization of the different body cells. Okay, so regulating gene activity allows for specialization of cells within the body. All right. So the way that this is going to happen is that certain genes are going to be turned on and off. And again, when something's turned on, that means it's going to be transcribed and translated. And when it's off, it's not going to be. Okay. And so first of all, just as a reminder, genes are going to determine the nucleotide sequence of specific mRNAs, and those, okay, so the mRNAs in turn determine the sequence of amino acids. I'm going to put AA for amino acids in proteins. Okay. So let me just write this out again. So gene turned on. This means is being transcribed and translated. Okay, so when you have your gene, it's turned on. So we'll say like it's turned on. It will then be able to be turned into mRNA, and then that will be able to turn into a protein. okay? If the gene is blocked, it blocks all of this. So it basically does not ever lead to a protein, okay? And so that would be, so this would be on and this would be off. Okay, so this whole idea is going to be called gene expression as well. So with gene regulation, gene expression. So certain cells are going to express certain genes. Okay, and so for example, we can look at this. So what we have here is, this is a pancreas cell. So this is going to be in your pancreas. It's going to be involved in insulin and glucose regulation. This is a white blood cell, which is part of the immune system, and so it's going to have important functions in defending your body against foreign invaders. And this last one is a nerve cell, and so the nerve cell is going to be important in conducting nerve impulses, so how you're able to touch, see, things like that. Okay, now if we look down here, we have a series of genes. We have a gene for glycolysis enzyme, so an enzyme that's required for glycolysis to take place, which, if you remember, this is all about cellular respiration. This next one is an antibody gene. We then have an insulin gene and then a hemoglobin gene. If we look then at the pancreas, we can then say, well, the pancreas expresses some of these genes, but not all of them. So the pancreas one is going to have the gene for glycolysis. It does not do anything with the immune system, so it's not going to do this antibody gene. It's going to produce insulin because that's one of its main jobs, and it also is not going to do this hemoglobin gene, okay? The white blood cell is part of the immune system. It does do glycolysis, and it also is going to have an antibody gene because antibodies are very important in the immune system. But then it's not going to have anything to do with insulin or hemoglobin, and so those genes are inactivated in that cell. This nerve cell is only going to have the genes for glycolysis of these four. Obviously, there's going to be many different genes that are going to be activated in the nerve cell, but from this list, the only one is actually going to do is going to be this gene for glycolysis. Every cell in the human body is going to have to be able to do glycolysis because this is how everything is alive. So you have to be able to make ATP in order for things to stay living. And so glycolysis is going to be one of these genes that's going to be activated in every single cell in our body. Okay? And here's just another little image of this. So we have, for example, the liver cell, and here's a neuron. So liver cells, one thing that they do is they have this enzyme called alcohol dehydrogenase, and it's able to kind of take alcohol and break it down into a form that's not as not toxic to our bodies, so it kind of breaks it down. So in the liver cell, you're going to have the gene for alcohol dehydroxygenase gene is going to be present, and the neurotransmitter gene that we would see in a neuron would not be present, okay, because this isn't going to transmit nerves. Neurotransmitters are something that are found in neurons. They are going to transmit a message from kind of one nerve to another. And in that case, we're going to have the neurotransmitter gene turned on, but this alcohol dehydrogenase gene will be turned off in the neuron. Okay, so both of these are going to have the gene for both of these different neurotransmitters, alcohol dehydrogenase, but they're not both going to be actually turned on. So that is going to be this kind of gene expression that's going on. So I wanted to just kind of answer a question right here. So if your blood cells and skin cells have the same genes, how can they be so different? Okay, and so the main thing about this is that they're going to express different genes than each other. Okay, so each cell type expresses different genes than the other cell type. Okay, so red blood cells, they're going to be built so that they can hold oxygen and transport oxygen from one part of our body to the other. Skin cells are going to have other types of functions. They're going to be important for protection and kind of try to prevent water loss and things like that. And so the genes that are going to be active in these two different types are going to be very, very different. All right, so to start talking about how this actually happens in cells, we can first look at bacteria. Okay, so we're going to talk about gene regulation in bacteria. Okay, so bacteria are single-celled organisms, and they're less complex than a multicellular organism and how they're doing their gene regulation. And so it's a kind of an easy way to start this introduction and how genes are actually regulated. Now, first, what I want to just tell you is that natural selection has favored bacteria that express only genes whose products are needed by the cell. Okay, so a bacterial cell is not going to want to produce a bunch of products or proteins that it's not actually going to be able to use. And so it needs to be able to regulate when to produce certain proteins, certain enzymes at different times. Now, this is the first time we've talked about natural selection, and so we will get into evolution very soon. But before we kind of get into that, I just wanted to mention, what does natural selection mean? And so natural selection, so this has to do with evolution, is the process whereby organisms better adapted to their environments tend to survive and produce more offspring. So what this means when it comes to bacteria is that bacteria that are only producing the genes and the gene products that they absolutely need are going to do better than bacteria that just make every single gene regardless if they need it or not. Bacteria that are not regulated and aren't regulating their genes are going to use a lot of energy and probably suffer and not do quite as well as bacteria that have really tight regulation and are able to be energy efficient and can survive better. Okay, so let's give you an example of gene regulation in bacteria. So this is actually something that happens. So we're going to talk about E. coli. And E. coli lives in your intestine. You can get food poisoning from E. coli, but E. coli is naturally kind of present. And so it's going to be living in your intestines. Okay. And so what happens is if you drink a milkshake, okay, so set up the situation. You drink milkshake or any type of, you eat ice cream, any type of thing where you're going to have a lot of sugar, lactose. Okay, so this is going to cause a sudden rush in lactose. Okay, remember that lactose is going to be a milk sugar. What happens is that the E. coli will express 3 genes for enzymes that enable bacteria to absorb and digest lactose. Okay. However, E. coli only does this in the presence of lactose. So lactose has to be present before they start to express these three genes. Okay. Once lactose is then gone, so after lactose is gone, so it kind of goes through the body, the genes are turned off. Okay? This is really important, and this is how our bodies work as well. So if there's some type of product that needs to be made, our bodies can make it, but then once we have enough of it and don't need it anymore, we stop making it, okay? And so this is gonna be kind of an important point. So how does this actually work? And how does the presence or absence of lactose influence the activity of genes, okay? So this is kind of a well-studied example that I'm gonna go over. Okay. So let me just write this. How does the presence or absence of lactose influence the activity of the genes? Okay. So first of all, the three lactose digesting genes are all right next to each other. So if you look at the DNA and the area where these lactose genes are, they're all going to be right next to each other, one next to the other. Okay? And because they're so close to each other, they can be turned on and off as a unit. Okay? Now, in addition to these three genes that are on the actual DNA, there are going to be other sequences of DNA that are going to be sites of regulation. Okay, so we're going to have short stretches of DNA near the genes that are sites of regulation. Okay, and so they help coordinate the activity of the genes. Okay, so what these are called in bacteria, and they're going to be called operons. Okay, and an operon is going to be a cluster of related genes and sequences, DNA sequences that control them. Okay, the one that we're going to talk about with lactose is called the lac operon. Okay, it has to do with lactose. Okay, so these DNA control sequences are going to have some names. So again, these DNA control sequences, the sequences themselves do not code for proteins, but they're involved in protein synthesis because they're going to either block or allow for RNA polymerase to bind to the DNA and transcribe something, and ultimately then that can be turned into a protein. Okay, so we have some things called the promoter. Okay, and so what this does is it promotes transcription. Okay, and what it's going to do is it's going to be a site on the DNA where the RNA polymerase attaches and initiates transcription. Okay, there is a second sequence, DNA sequence, that's called an operator. Okay, and this is going to sit in between the promoter and the lactose genes. Okay, what the operator does is it acts as a on-off switch. Okay, and so if something is bound to the operator, it's going to then turn it off, and when there's not anything on the operator, it's turned on. Okay, and so it's an on-off switch. I guess I should finish this. Depending on whether a specific protein is bound there, so to the operator. Okay, so if an operator is bound, or sorry, if, I should have said this, if a protein is bound to the operator, so let me just start that over because that's all backwards. So if a protein is bound to the operator, RNA polymerase, which I'm just going to put RNA pol, cannot bind. So it's repressing that. Okay? This protein that we're talking about right here, the protein is called a repressor. Okay, so it's going to make it so that the gene is not going to be allowed to be transcribed. Okay? So I want to show you what this kind of looks like. And first, just want to draw you what this actually, kind of a very simplified version of this, and I'll show you a little more complicated version. So we have a sequence of DNA, and part of it is going to be what we call the promoter. We just talked about that, promoter. Okay, so there's this little sequence of DNA right there that's a promoter. Then we have a different sequence right here, which is going to be called the operator. Okay, and remember, this is the on-off switch. And then right here is going to be those 3 lactose digesting genes. Okay? So what happens is that the RNA polymerase will bind here, and this is where the repressor can bind. If the repressor binds onto the operator, it means that we can't proceed. Okay, so like a repressor will come in here, kind of sit on top of the sequence, and the RNA polymerase can't kind of move forward because this is going to block it. If the repressor is not there, then you can have that sequence actually transcribed. So this is what this looks like. So we have our DNA, we have a promoter in the green, we have an operator in the yellow, we have this lactose genes. This whole entire thing is called the operon. And we have this protein, which is going to be a repressor, okay? So if the repressor is active, and the only time it's active is when there's no lactose. So when the repressor is active, the repressor will actually bind to the will bind to the DNA at the operator site and make it so that the RNA polymerase cannot attach. Okay, so how do we actually start to make these genes for lactose? The way that they're made is that we have to actually have lactose present. So when lactose is present, it will actually bind to the repressor and the repressor becomes inactivated. Okay, so the presence of lactose inactivates the repressor, which allows RNA polymerase to bind to the promoter and can then transcribe the mRNA, which is going to make the lactose enzyme. Once we have the lactose enzyme, it's going to start to degrade lactose. Okay, it's going to break it down into glucose and galactose, and it's then going to make the lactose amount drop. Once it drops enough, it will then go back to an active state and will start repressing the production of lactose. You do not want to have lactose enzymes present unless there is actually lactose there. Okay, otherwise it's going to go to waste and this is expensive to make it. So this is how this lac operon is going to work. Now, there are multiple different types of operons that have found in bacteria, and they all work very similar in this way. All right, so now we're going to move on to eukaryotes, because eukaryotes, which these are going to mostly be, the ones we talk about are mostly going to be multicellular. They're going to have a much more complex type of regulation, okay, because there's a lot more going on. So we're going to start talking about eukaryotic gene regulation. Okay, so eukaryotes are going to be often multicellular, not all of them. Okay, and they're going to require more sophisticated mechanisms than bacteria for regulating expression of genes. Okay, and basically we've talked about the central dogma. So we have kind of DNA to RNA to protein, and we also have transcription, translation. Basically, this whole pathway can be regulated at every single step in multiple different ways. Okay, so we can regulate the actual DNA and make it so that it can't be transcribed, or it can be transcribed. We can then affect transcription. We can promote it or make sure that it doesn't happen. The RNA can be processed or it can be unprocessed, or modified in ways that are going to change the expression of the genes. And then translation can also be regulated, and protein. Okay, and so we're going to talk about the different steps of how this actually happens and what things are actually being tightly regulated. Okay, so the different types, we're going to go kind of give a brief description of some of these different types. So the first one is going to be called chromosome availability, or accessibility. Availability actually works as well. And so what's going to happen is that the chromosomes themselves can be structured in a way that makes them easier or harder to transcribe. Okay, so the structure of the chromatin. Okay, so remember, DNA is going to be both, chromatin is both DNA and proteins. So we talked about histones. So the chromatin can be regulated. Okay, so if you have kind of a looser chromatin structure, you're going to have more transcription take place. Okay, so we'll say this: more open chromatin, so not quite as tightly packed, makes genes more available for transcription. Okay, if you have more closed chromatin, it's going to make it more difficult for transcription to take place. And it's kind of like, how tightly have you wrapped your DNA around those histones, and make it easier or harder to open. The next one we're going to talk about is going to be transcription regulation. This is probably the most important one. So this is going to be the key regulatory point for many genes. Okay, and we're going to talk about these things called transcription factors. Okay, that will bind specific DNA sequences. Okay, and then they can promote or repress transcription. Okay, we're also going to talk about enhancers and silencers. So basically we have these different ways to promote transcription, enhance transcription, or repress it and silence it. The next step is going to be RNA processing. Okay, and so during RNA processing, we will splice, cap, and it's the addition of the poly A tail. Okay, this can be regulated. So the RNA can or cannot leave the nucleus. Okay, if the RNA is to leave the nucleus, it absolutely has to have capping and that poly A tail. So you can regulate whether that actually takes place. Okay, so we're going to talk about different mRNAs can be made from the same pre-mRNA by a process called alternative splicing, and I'll talk about that. Okay, so we'll go into more detail about that. So you can affect whether the capping in addition to the poly A tail or not takes place, but then also splicing can be regulated, and different proteins can be made by alternative splicing of an mRNA. The next is going to be RNA stability. Okay, and so the lifetime of an mRNA in the cytosol, so once it leaves the nucleus, affects how many proteins can be made from it. Okay, so if you have an mRNA strand that's present for a long period of time, a lot of protein can be made from it, but there are other times where you don't want it to make very much protein, and so you would have to destroy the mRNA strand. Now, we'll talk about a couple different types of molecules that are actually going to be involved in this RNA stability, and some of them are going to be what we call miRNA. And these are going to basically bind and destroy mRNA. Okay? Translation is also a site of regulation. Okay, so you can increase or inhibit translation. Okay, and there are going to be regulators. We don't really talk about translational regulation much, but there are different proteins that are going to be involved in this. And then the last type of regulation is going to involve the protein itself. Okay, and so once the protein is produced, we can change its activity. Okay, so proteins can undergo a variety of modifications. Okay, they can be chopped up and destroyed. They can be tagged with chemical groups. Some proteins actually have to be modified before they'll even be functional.